All right, y'all. Renee Kazishki here, joining you from my dining room, which right now looks like a bit of a disaster. Got some art projects and files and jackets and disaster in there. But look, I just want to show you this. This is my window. We cut out some hearts. And I'm talking in this voice, and it could be annoying to you, but it's entertaining to me. And sometimes it makes people pay attention because they're not quite sure what's going to come out of this version of Kazishki. So this is going to be a review of Chapter 6 and 7, which corresponds to Unit 2 in the College Board's curriculum, basically sales and cell transport. I'm going to try and move quickly and hit the big ideas. You can watch the College Board videos for their interpretation. My thumb is a little weird looking, so you're going to see that. But here we go. Prokaryotes versus eukaryotes. Two major cell types, biggest division on the living planet until we found that prokaryotes actually exist in two domains, the bacteria and the archaea. So prokaryotes have a lot of diversity in between these two domains, but the thing that they have in common that makes them distinct from eukaryotes is they don't have a nucleus, they're extremely long-living here on this planet, and they're very small, they're simple, they're not very organized. Now when we say small, simple, and not organized, we think that means dumb, and they are dumb. They are extremely successful, like the most successful living thing on the planet. All right? So don't forget about how good they are at reproducing, how good they are inhabiting all different kinds of environments, how good they are at just evolving, how their fast reproduction rate, their pretty generous mutation rate has allowed them to exist for billions of years longer than eukaryotes. All right, now eukaryotes. The feature that eukaryotes have that prokaryotes don't is they are compartmentalized. What that means is this is like you're living in a one-bedroom, no, sorry, not a one-bedroom, a zero-bedroom apartment. You just got a bed, maybe a sink, if you're lucky, a semi-kitchen, and it's all in one room. Compartmentalized in the eukaryotes, you're living in a multi-room household. You got places to do stuff, places not to do stuff. For example, you got a place to make ATP energy in cellular respiration. And if you're a plant or an algae or photosynthetic bacteria, you also have, well, actually the bacteria don't. That'd be over here, my bad. That was a big slip. They'd be over here. But if you have chloroplasts, you got a definite spot in which to do that. And it's not going to mess up with your other stuff. All right. Endomembrane system, that is something I thought needed a little elaboration. So I had this picture here for you. Got eukaryotic cell, DNA in the nucleus, form a chromatin, nuclear membrane, cell membrane. The endomembrane system involves, first of all, getting a little piece of mRNA, processing it, right, before it leaves the nucleus. So this is going to be the primary transcript. And then we have the mature mRNA. And then that is going to attach to a ribosome, boys and girls. But the ribosome, if it's part of the endomembrane system, is going to be on the rough ER. There we go. Oh, there we go. There we go. Oh, let's do one more. There we go. And the rough ER is going to be rough because it has ribosomes. So here we go. Mature messenger RNA attaches to a ribosome on the rough ER. And what happens? Does the protein that's translated from that just float around in the cytosol? No. It's released in a vesicle. So that P represents new protein, all folded and ready for the next step, which is it's going to leave the rough ER. in its little vesicle. And then it's going to enter something called the Golgi apparatus. And it's going to go through the Golgi and be modified. What do we mean by that? Golgi modification is like putting some accent beads on your necklace or something like that. It's like chemically altering the protein, making it a little bit different on its ends, perhaps adding some functional groups, just making it more special. And then from the Golgi actually decides, and no one knows what that means, but decides where it's going to go. So this could be in a lysosome. So this could be a protein in a lysosome, meaning it's got some hydrolytic properties, going to keep it locked up in that vesicle so it doesn't destroy the rest of the cell. And now it's there. Or it could be a protein that's stored in a vacuole. All right? Or that protein could go to the membrane and be expressed on the surface of the membrane, like be a receptor or something, like for cell communication. Or that protein could get exported. So this would be like membrane bound, so like a receptor or something, and this would be a protein that's exported. Basically, with the endomembrane system, you got compartmentalization. You got things happening in little bubbles, in little areas. They're not disorganized, they're very organized, and you have protein synthesis happening from the DNA in the nucleus in conjunction with the rough ER through these vesicles that move and then become different things based on what that protein is. All right. That was a little bit long, but now we're on to number 2. We're going to take a break in a minute. Cell diversity in a multicellular organism. This is kind of out of context because it's more about Unit 6. Cell diversity is due to differential. Now, if we in class, I'll be playing hangman with you. Gene expression. All right, boys and girls, you know what that means. That means that different cell types take on different looks, properties, functions, abilities, skills based on the genes that those cells are using. So if you're a muscle cell, you're going to use different genes than if you are a different cell. So this is about multicellular organisms and why cells look so different from each other. They have the same genes. They all do. They're just using different ones, expressing different ones. All right, last topic, then we take a break. Cell size. When we look at cell size, we calculate a ratio. It's the surface area, which represents the membrane or the amount of membrane, compared to, in this ratio, the volume, which represents the cell's needs. And as a cell grows, this number goes down. Okay, don't forget this. As a cell grows, surface area to volume rate, surface area to volume ratio goes down. So, small cells have a high surface area to volume ratio. What that means is they have a lot of membrane to be able to meet relatively small amount of needs. As a cell gets bigger, they have less membrane compared to their needs. They meaning cells. All right, so I can do some sample calculations. I don't think it's really necessary. It's just a reminder, small cells have an advantage. It's why you're made of trillions of cells instead of like 5 basketball cells, basically. We're going to come back in the next video with membrane structure and function. This will correspond to Chapter 7 and Part 2 of this little fun activity. Miss you guys. Hope you got something out of this. I sure did. Peace.